First of all, hello everyone, and thank you so much for allowing me to present this work. So I will be talking about imputing links L1000 drug signatures, and I am a PhD student under Joel Dudley, who's, I think all of you guys know, he's co-director of the Mount Sinai KMC. And this work is in collaboration with some folks over at IBM TJ Watson Research Center, as well as with Avi Mayan and a couple of members of his lab, and under the guidance of both of my advisors, David Sontag at NYU and Joel Dudley. And before I jump into this project, I wanted to talk a little bit about kind of a broader context. This is work that's going towards my thesis, and so I wanted to kind of explain to you guys my broader vision for using the links L1000 data in a kind of drug discovery platform. And so it's all kind of jumping off of this idea of drug repurposing via signature matching. And so I imagine that some of you are, I know that some of you are familiar with this concept, but I'll go ahead and walk you through it. Okay, so as I was saying, I'm kind of jumping off of this idea of drug repurposing via signature matching. And so the idea is that you can generate drug perturbation signatures, such as the ones in the links L1000 data, where you compare gene expression between untreated and treated samples for treated with various chemical perturbations, and that generates an expression signature for multiple drugs or chemical compounds. And similarly, you can generate an expression profile characterizing a disease by comparing expression data for diseased versus healthy individuals. You can do this for multiple diseases. And the idea of signature matching, which was originated by the Broad Institute 10 years ago, is to basically play this game of matchmaker, where you can compare any pairwise combination of a drug and a disease by looking at their expression profiles. And the idea is to identify kind of opposing expression patterns, with the rationale being that if expression is perturbed in a certain direction in disease, and a drug shows evidence of perturbing the expression in the other direction, then perhaps that drug could push the expression, the disease-perturbed expression, more towards normal, and thereby providing some sort of therapeutic benefit for that disease. And so a lot of work has been done using this approach, and maybe one of the most cited papers is the one I'm showing here, that led to two unintuitive drug-disease connections. For example, using topiramate, which is originally an anticonvulsant for IBD. Okay, and so here's a more abstract view of signature matching, where you just have a drug profile and a disease profile, and you compare them, and this leads to some drug repurposing hypotheses. And I came across this idea early on in my thesis, and was really kind of enamored by the idea, because it's quite simple and sort of just very mathematical, and I come from a mathematical background. And yet it leads to very data-driven and sometimes unintuitive hypotheses that seem to be working. On the other hand, this approach, when I think about this approach, I think about all of the other data surrounding the drug space, if you will, and the disease space, and so I'm asking the question, can we improve methods for drug repurposing and other sort of pharmacological aims by integrating data across drugs and other dimensions? And I'll give you a picture to be more specific of what I'd like to do. So I would like to transform this picture into something that looks more like this. And so at the center, I have this kind of cube of expression data, some sort of multi-aspect expression data. For example, one dimension would be genes, and then I could have another dimension being multiple drugs, and another dimension being different doses or different cell types, etc. And the idea would be to really kind of integrate these data together and kind of model the relationships between these different dimensions, and also integrate this with additional information such as chemical structure, gene pathways, etc., and not only generate drug repurposing hypotheses, but also hopefully be able to predict drug-target interactions, predict, elucidate drug mechanisms. So this is kind of the big picture. And so the project that I'm presenting today is really focusing on looking at the relationships between drugs and cell types in terms of the gene expression space. And so just to reiterate, my goal is to model the relationships between drugs and cell types in the Lynx L1000 data. And the motivation to do this is twofold. First of all, the one that I'm focusing on today is to impute missing signatures in the data. And second of all, I would like to identify unexpected patterns in the data. I'll talk a little bit more about that at the end. And so why would we want to impute missing signatures? Well, I've come up with 3 reasons. So first of all, we could use these imputed signatures and just treat them as if they were measured, and so you could do downstream analyses such as the drug repurposing that I just outlined. Another reason is that by imputing the data and kind of filling out the space, this would provide a common set of high-dimensional features for each drug. So in other words, if I were to fill in the space of all drug cell combinations, then for each drug I would have the same set of features being an expression signature for each of the different cell types. And so that would enable kind of easier machine learning, kind of building on top of that to make predictions about those drugs. And then the third motivation is that of why I'm doing this is that it could apply to other settings beyond the Lynx L1000 data. For example, if I modeled the relationship between individuals and specific tissues, I might be able to impute in vivo brain expression, whereas that's something that you wouldn't be able to measure. And so this could basically enable you to predict gene expression or other sort of high-dimensional data in situations where it may not even be possible in the first place. So the outline for my talk is, first I'll just talk about the particular sampling of Lynx L1000 across kind of drug cell combinations, and then I'll talk about tensor modeling of Lynx data, and then I'll talk about some results. And then I want to talk about how these imputed signatures might actually be applied, and then some next steps. So first of all, the sampling. So here's a view of this 2-dimensional space of cell types versus chemical compounds. And so the Lynx L1000 data covers 77 different cell types, and although Lynx has actually generated, the Broad has generated measurements for over 20,000 different chemical perturbations, what I'm showing are perturbations that have been deemed to be significant at kind of like an overall expression perturbation. So just nominal significance of p-value less than 0.05, and that analysis was done by Avi Mayan's lab. I'll talk a little bit more about that in a second. But anyways, what you see here is that the sampling is very, it's rather sparse for most cell types. While there's about a dozen cell types that have a fairly large number of compounds in which they've been measured, but in most cell types there have been very relatively few compounds. And so I'm actually, what we've done so far is just focused on this fairly highly sampled space in the top left corner delineated by the red box. And but the hope is that we can then extend the analysis to be able to make predictions in this larger space. Okay. So now I'll talk about tensor modeling and how I kind of bring links L1000 into the tensor framework. So what is a tensor? A tensor is a multidimensional generalization of a matrix. So what I'm showing on the slide is a three-dimensional tensor over genes, drugs, and cell types. And you could have even, 4 or 5 or n dimensions of a tensor. And there's this notion of a low-rank tensor approximation that would be analogous to low-rank matrix approximations, such as the SVD, which is also it's another name for PCA. And these low-rank approximations capture linear relationships between the variables. And so the idea is we want to kind of we're going to model the relationships between drugs and cells as they affect genes in a linear way in order to be able to fill in the missing entries. And so this is the particular tensor that I've constructed so far. And so the idea is I want to populate the tensor with differential expression profiles, one profile for each drug-cell combination, kind of in this red box here. And I'm using signatures that were generated by Avi Mayan's lab, and they applied the characteristic direction to compute differential expression. If you guys haven't looked into this and you're doing differential expression, I do highly recommend it. And they, as I mentioned earlier, they they came up with a notion to kind of evaluate the overall expression perturbation. And so I just wanted to filter out signatures for which I was reasonably confident that there was not a strong signal. And I selected a subset of 746 drugs and 11 different cell types that had a higher observation density. And the cell types are shown on the right here, as well as the number of signatures that I had. Any questions so far? Okay. And so what I end up with is a tensor like this, where I have columns that are missing. So I'll have a particular drug-cell combination that does not have a measurement or for which the measurement was not significant. And so what I have is this tensor where either the column is present or it's not present. And I'm going to apply tensor completion to this tensor. So this is analogous to matrix completion, if you've ever heard of that, a kind of famous version of matrix completion is the Netflix problem, where you had different users and movies, and you had some entries of that matrix filled in, identifying which users liked which movies, and then the problem was to complete the matrix in order to predict what other movies each user would like. And so this is a similar idea, except in higher dimensions. And so the idea is to impute missing entries based on the observed entries. And certainly you're going to have to assume some sort of relationship between all of the data. Otherwise, it would be impossible to predict what the missing entries are. And so all of the tensor completion algorithms that I'm aware of assume some sort of low-rank structure. And another application of tensor completion would be video or image reconstruction. Okay, so I'll talk about the cross-validation setup and the results. So, I ran a cross-validation experiment in order to evaluate if this would actually work on the L1000 tensor. And so this is how I set up the cross-validation. On the left, you see the tensor with the original missing data, as well as a single held-out signature. And then I can run tensor completion, and I get all of the missing data. I get imputed values, and then I can just compare the imputed signature versus the held out signature with some measure of distance, such as cosine distance or L2 error, like Euclidean distance. And then I also used a baseline for comparison, which is simply the average expression across cell lines per drug. Okay, so if I had 5 measured signatures for that drug in different cell lines, I would just compute the average and fill in the remaining missing entries. And in terms of the tensor completion algorithm, I used something called FALRTC, which stands for Fast Low Rank Tensor Completion, which was selected among other algorithms after some benchmarking. I won't go into the details. And so here are some results of the accuracy when I ran a leave one out analysis. So in other words, I reran this process, holding out one signature at a time. And I did this for every possible signature that's in the L1000 tensor. And then I looked at the results. So on the y-axis, I have the imputed values, and on the x-axis, the true values. And each dot, so this is a smooth scatter plot, but each dot represents basically a single element of the tensor. So it's a single voxel or gene in a particular drug cell combination. And of course, what we're looking for is a slope of 1 and a correlation of 1. And what I ended up with was both a slope and correlation of 0.69. And next I wanted to look at some ROC curves. So basically, I wanted to evaluate whether the imputed signatures were able to identify the same genes as being differentially expressed. And so what I did is I took each signature and defined some true set of differentially expressed genes just based on some threshold. And then I took the imputed signature and I varied the threshold to generate a kind of varying set of differentially expressed genes. And so by varying the threshold on the imputed signatures, I generate ROC curves. And so what I'm showing are results for both the tensor method, which are the solid lines, and then the mean baseline, where I just average the expression data, and that's shown in the dotted lines. And the difference between the blue and the red is just where I draw the threshold to define the true differentially expressed genes. So the blue curves correspond to a stricter threshold to identify only 0.5% of the genes as being differentially expressed, whereas the red shows a lower threshold of roughly 9% of the genes being differentially expressed. And so what I see is that the tensor approach does perform better than the baseline in both cases. And I do observe that in general, the more strictly I define differentially expressed genes, the better both methods perform. And this is kind of as you would expect, because it's also a good sign, because what we care about are kind of the most extreme values in the tensor, or the genes that are giving us the strongest signal. And so what this is saying is that both methods are better at identifying the genes that have the strongest signal versus genes that have sort of a, less strong signal. Any questions so far? Okay. And so my next question was, does the imputation performance vary per gene? And so what I did was I generated similar ROC curves to this one, except I did this only looking at one gene at a time. And then I computed the area under the curve. And then I did this for both the tensor, the solid line, and the mean baseline, the dotted line. And so what I'm showing here is a scatter plot of those AUC values, those area under the curve values. On the y-axis is the tensor approach, and on the x-axis is the baseline approach. And so an area under the curve greater than 0.5 means basically better than random, and the maximum value is 1. And so what we see is that, and so the diagonal line basically would correspond to the tensor approach doing having the same performance as the baseline approach, and above the diagonal means that the tensor approach is doing better than the baseline. And so what we see is that at a gene level, for almost every gene, the tensor approach does do better than the baseline. And another kind of immediate observation from this plot is that the performance does vary quite widely per gene. So some genes are showing results near 0.5 for both methods, and some genes are showing results close to 1. Okay. And another thing that I noticed is that some genes perform, the tensor approach performs much better on some genes than others, in particular these genes up here. And so I wanted to see if there were any patterns that I could identify to help me understand, where exactly is the tensor approach performing well, where is it kind of lacking, and so I'm hoping to analyze this in order to identify, how to move forward. And so I wanted to, analyze which genes I'm doing well on. And so I focused in on 2 different regions of the plot. So first of all, this yellow region here is showing the genes for which the tensor modeling performs better than the baseline. And then the orange region is just showing genes that the tensor approach does well on, regardless of how the baseline approach does.
Rachel, I apologize. Can I interrupt you? There are a couple of us confused over here. We don't recall what exactly baseline approach is. Could you say again?
Yes. So the baseline approach is simply computing the mean expression value across, so take a particular drug in the tensor and just look across the cell type dimension and average the expression values for all the measurements that I have in that dimension. Does that make sense? So in other words, the idea is that...
So for each gene, you calculate average expression levels across all cell lines for a given drug, right, for which you actually...
Yes, within the tensor.
Thank you.
Mm-hmm. And so, the idea, kind of the reason that we used that baseline is because without that comparison, we were just wondering, maybe that's all the tensor approach is doing. It's just kind of, looking across the cell lines and saying, oh, this is what I think the drug is doing in this cell line. So the baseline hypothesis, the hypothesis that the baseline approach is making is that a drug's effect on gene expression is not cell-specific. Does that make sense? And so the tensor approach is hopefully picking up on some more cell-specific patterns and kind of looking at the patterns across the entire tensor. So I'll continue to show you some of the Go biological process enrichments that I obtained for these gene sets. And so the first thing that you notice is that, okay, so the top enrichments that I'm showing, this entire yellow set, are the enrichments where are the genes for which the tensor does better than the baseline, and the 2 orange gene sets correspond to just genes that the tensor approach does better on, does well on in general. Oops. Okay, and so the first thing that you notice is that we found many enrichments for the first comparison and only a couple enrichments for the second comparison, although the enrichments are relatively strong in that second comparison. And so I don't have a whole lot of analysis to explain to you why I changed these particular enrichments, but one thing that I did notice, which goes along with my intuition, is that the tensor modeling performs the gene sets for which the tensor does better than the mean. There are a lot of cell-specific gene set enrichments, which I highlighted in red. Hopefully you guys can see that coloring. But you see positive regulation of endothelial cell migration. You have brown fat cell differentiation, osteoblast differentiation, neutrophil chemotaxis. So a lot of cell-specific processes which are enriched, and so that is definitely something that I find concordant with my intuition. If anybody has ideas about why the particular gene sets are enriched at the bottom, I would be happy. I would be all ears. But I'll go ahead and move on. And so, this basically concludes my analysis of the imputation that I've done so far. And I'll talk more about how I want to extend this work at the end. But the next thing that I want to talk about is some work that I've done to evaluate the utility of these signatures in a particular application, which is this drug repurposing via signature matching. And so, yes.
Before you continue, I have a question. So one of the things that you said, you told us that you removed the non-significant perturbations.
Yes.
So, and then you had your tensor.
Yes.
But if you then you try to predict those perturbations that were not significant, there is already an experimental evidence that the drugs, let's say, don't do anything to the cells. So how
Absolutely.
do you consolidate this concept, and is the tensor predicts that those drugs are not going to do anything?
Yes, I'm glad you asked that question, and I just started thinking about how we could incorporate that into modeling, because right now what I'm doing is basically throwing those signatures out and pretending that I never measured them. But you're absolutely right that we are throwing out that information that we, essentially we don't think that there's a significant perturbation that happens in that drug cell combination, and so I'm sure that there's a way to incorporate that into the modeling by sort of, maybe trying to impute the data where I allow zeros in the expression, in the expression signature, so that I can basically either choose to predict that the expression is non-significant or, predict a particular direction. So I would definitely like to incorporate that into the model. So as I just said, this next piece of work is just talking about evaluating the utility of the imputed signatures for drug repurposing. Because, so far I've kind of showed you some quantitative evidence that these signatures are reliable, or the degree of accuracy. But I think that until I can show some sort of biological concordance, or sort of use these imputed signatures in different applications, I think that's when I can really begin to convince people that what I'm doing is actually useful. So, the question that I'm asking more specifically is within this framework that I presented at the beginning of signature matching. Okay, and the question is, would my imputed drug signatures connect to the same diseases relative to their measured counterparts? And so I set up an experiment. So first of all, I needed some disease signatures to compare to my drug signatures, and these were curated by Avi Mayan's lab, along with the links BD2K, DCIC consortium. And so these signatures were extracted from GEO. So these were crowdsourced, and they've generated about 900 disease signatures to date, and 589 of them overlap with the L1000 landmark genes. And they are represented as sets of up and down regulated genes with numeric values coming from the same characteristic direction analysis that was applied to the drug signatures. And they are available for download after you contribute at least one new disease signature. And so I set up this experiment where I wanted to, this is what I did. I applied the signature matching technique to compare all of these disease signatures to all of the original measured drug signatures first, okay? And so what I end up with is this matrix shown on the slide of diseases by drugs, and I'm just keeping things at a cell-specific level. And I have a value that compares these two signatures. I'm using cosine distance. So what I'm looking for is opposing directions of signature, and so higher cosine distance is better. Okay, and then step 2 is that for each disease, I'm going to find the top ranked drug signature, remove it from the link's L1000 tensor, and impute it. Okay, and then I'm going to rank the new imputed signature against all of the other measured drug signatures and see if it remains in the top ranks. And so then my new rank, going down to step number 3, the new rank of the imputed signature is what I'm using as a proxy for quote-unquote drug repurposing error, since ideally the new rank would remain as 1. And so let me just try to reiterate this process in a little bit of a simpler way, since it's important to understand the results. So in other words, what I'm doing is I'm finding the strongest drug-disease connections, and then I'm replacing that drug signature with an imputed drug signature and seeing if that connection remains strong. And that new rank is how I'm measuring that strength of the new connection. And so here are my results. So I did this for, I just rewrote the same line on the next slide. The new rank of the imputed signature is a proxy for the drug repurposing error. And so these are the results on 40 different disease signatures that have the strongest connections to drug signatures. And so what I'm showing you here is the distribution of the new ranks. So again, the ideal would be that the entire distribution is at a new rank of 1. And I found that the median rank is 20 out of where the worst possible rank would be 6,055. And the mean percentile was that these imputed signatures remained in the top 1.2%. The best case in 2 out of 40 of the tests, the imputed signature remained as the top comparison, the top hit for the disease. And in the worst case, the signature fell to a rank of 784, which is in the top 13%. And then finally, 15 out of the 40 comparisons remained in the top 10. And for me, maybe that last one is the most interpretable, in the sense that, if I were actually doing a drug repurposing study where I had a particular disease signature for which I wanted to prioritize drugs, I think it's quite reasonable to, have some assay for which I could test 10 compounds. And so if, that the drug that I would like to prioritize remains in the top 10 with the imputed signature, then I would not have lost it.
I'm curious, have you tried this with baseline signatures?
I have not, actually.
Okay.
Yeah, I should. Thank you. Okay. Any other questions? Okay. And so the last thing that I'm going to talk about is kind of continuing on in the same vein. I wanted to see if the imputed signatures would actually generate new hypotheses. And so the goal was to find disease signatures where the best match was an imputed signature. And for the 589 disease signatures that I tested, I found 63 examples of this. And so here are some of the, these are the top, I think, 20 of the 63 examples that I found, excuse me, connecting drug-disease pairs. And with a quick literature search, I did find some support in the literature. So, for example, there was a prediction based on one of the imputed signatures connecting paclitaxel with idiopathic pulmonary fibrosis. And indeed, there's a paper about low-dose paclitaxel treating pulmonary fibrosis. In addition, chelidonine treating atopic dermatitis. This is one of the known uses of chelidonine. And then finally, there's another prediction for this BIBU 1361, also for treating IPF. And I did not find a particular paper talking about this connection, but what I did find is some talk of the epidermal of EGFR being important in idiopathic pulmonary fibrosis, and then it turns out that this drug BIBU is an EGFR inhibitor, and so maybe there's something there. So so it looks like these imputed signatures are actually doing something reasonable and might really actually be useful for downstream analyses such as drug repurposing. Okay, and then finally I just want to talk about some next steps for this work. So first of all, I would like to analyze the residuals or the errors in more depth. And so here's an analogy of why I think it's important. So near the beginning of the talk I said that one of my motivations for modeling the drug-cell relationships was to identify surprising patterns in the data. So here's an analogy of how this might work. And so what you see here is an application of matrix completion in order to reconstruct this image. So the original image is on the left, and then they add a bunch of noise to the image, which I'm not showing, and then they reconstruct the image on the right. And what you see is that overall, the reconstructed image does a quite good job of generating this image of the building, but some of the kind of idiosyncratic features in the image are not very well captured in the reconstructed image. And so if you imagine looking at the difference between the original and reconstructed image, then suddenly we would see larger residuals in those areas, and that could kind of guide our analyses to more interesting places. My hypothesis is that a similar thing would happen in the Links L1000 data. So for example, I might identify particular drug-cell combinations for which the drug is doing something very nonlinear or unexpected in that particular cell type, and that might reveal some kind of interesting mechanism that's specific to that cell. And then also next steps, we plan to experimentally test some of our predictions very soon. And then kind of farther down the road, I would like to extend this analysis to larger regions of the L1000 drug cell space and incorporate significance of expression perturbation. That's basically addressing the concern that Avi raised. Also, I'd like to provide confidence intervals for the estimates and extend to nonlinear modeling. And so with that, I'll conclude. I'd like to acknowledge my advisors, Joel and David, as well as members of Joel's lab, Ben Reedhead, Brian Kidd, Milo Smith, Lily, Amy, and Roman, members of Avi's lab, performing the differential expression analysis, Xiaonan, Neil, and Avi, IBM, and the Broad Institute. Thank you. And I'll now open to any questions.
You didn't give any detail in the algorithm that you use for imputation, but I was wondering whether in the imputation process you use in any way the molecular data that are available for every cell line, like the copy number variation or mutation or even the base expression in every cell lines that I imagine would be able to drive the imputation process better. Thank you.
Okay, that's a great question. Thank you for bringing that up. The answer is, so first of all, I can send you the paper of the imputation method that I'm using. And to answer you can just shoot me an email afterwards. In terms of the second question, incorporating additional information, the answer is no, I'm not doing that yet, and I should have included that in future work. That's definitely something that we'd like to incorporate, especially if, you know, I think that this would enable us to not only predict kind of, you know, within the measured space, but also predict beyond into other, you know, cell types, for example. This imputation approach, I could definitely imagine it being applied to other, you know, high-dimensional data sets. Basically, you know, if you have a data set which can be constructed into at least a three-dimensional tensor, you would be able to, it's conceivable that you could use this approach to impute data or, you know, identify, you know, significant versus non-significant perturbations and things like that. You know, there's a lot of different variations that you could imagine.
One thing that I want to just, it's a comment that in your exam, in your proposal exam that I liked is the term that you used to describe the validation that you used, and you called it PubMed validation. And that's basically after you make those predictions going down the list and looking for low throughput evidence. And that's something that we do a lot, and that's like sort of like a way to validate your data using external sources. And it's like, it's funny, sort of like you coined that term that it's called like PubMed validation. So I thought that was pretty cool.
Right, yeah, proof by PubMed.
Proof by PubMed, yeah.
And maybe that's, I don't know if Joel originated that, he uses that term a lot.
Yeah.
But yeah, I mean, it's a place to start.
I know you're offering a paper about this tensor completion algorithm, but can you summarize it in just a couple of sentences to give us an idea of what's going on?
Sure. So, okay, actually, let me show you. I can try to offer some intuition in terms of what a low-rank tensor approximation is in general, and then I'll talk more specifically about the algorithm that I used. Okay.
Great.
So first of all, I just want you to see this figure on this slide. So when I think of low-rank tensor approximation, I think of the simplest one, which is called a CP decomposition, and it's shown pictorially on the slide. And this is, so what you see is the tensor on the left, and it's approximated by some number of these rank 1 tensors. There's R of them shown on the slide. And the reason that this is a rank 1 tensor, this is an outer product of vectors, one vector for each dimension of the tensor. Okay, and so in other words, like so if I just considered one of these rank 1 tensors and I looked at the ijk element, it would simply be a product of like the ith element of vector A times the jth element of vector B times the kth element of vector C. Okay, and so it's a very simple relationship between the A, B, and C vector. And this is directly analogous to PCA, where this would simply be reduced to a two-dimensional version of this. Okay, and another way to kind of intuit about this is suppose that the A vector corresponds to the gene dimension. Okay, and so what I would have is for each of these factors, Can you guys see my cursor?
Yes.
Okay. Okay. So for example, in the first vector, suppose that this here is the gene vector, and so it has some continuous values corresponding one value for each gene, and so I think of that in a simplified way as a pathway. Some sort of set of genes which are upregulated, things like that. Okay, and so that's my simplified interpretation of that A vector, and then the B and C represent how much. So B could represent the drug dimension. So B is saying if drug 2 has a value of 0.5, that means that it's activating this quote-unquote pathway. It's multiplying it by 0.5. Whereas drug 3, it might be negative 100. So it's really really strongly down-regulating this pathway. And then independently, this model looks at cell types, and it says, okay, this cell type at a sort of baseline is affecting this pathway by multiplying whatever, cell 3 multiplies this by 0.2, whatever. And so you can look at this as kind of independent effects of drugs and cell types affecting each pathway. And I'm kind of decomposing the expression into multiple pathways. So any questions about that explanation before I talk about the algorithm? Okay, so then this algorithm, the fast low-rank tensor completion, it doesn't use this particular decomposition, but what it does is it tries to approximate the tensor using a low-rank tensor. So rank meaning like the number of these that I would need. And so it sets it up as an optimization problem to try to minimize the rank of the tensor subject to the constraint that the predictive tensor matches all of the observed values. And so, the way that they solve the optimization problem is kind of complex, but yeah, I can send you the paper. Does that help?
Yes, right.
Okay.
Okay, so you basically fit your model using this algorithm, right? And then you have your reduced representation, and this gives you the prediction of all other dots that are missing.
Right. So so here's a simpler algorithm than the one I'm using. I'll describe a simpler algorithm that can actually impute the missing values. Okay, and it goes like this. So start with my partially observed tensor that has some missing values, and just fill in the missing values with random numbers. Okay, then take that tensor and perform a CP decomposition, and approximate that tensor using some low rank number of factors. Okay, once I have that approximation, I can then use that approximation to predict the missing values better than the ones that I had before. So I can replace the missing values with those predictions and then basically rinse and repeat. So that's kind of just to give you some intuition about how one could possibly do tensor completion. But it turns out that that approach doesn't work very well.
Thank you everybody for joining us. We'll post the recording of Rachel's presentation shortly on YouTube, and provide the links on a DCIC website.